Bjorn, I'll thank you. And Alex, I would like to hand it over to you to present the analysis tools. Thanks very much.
Yes, of course, no harm in being ahead of schedule. And Bjorn already introduced a little bit the concept of analysis tools. So what analysis tools are, as opposed to prediction tools where you try to predict what the particular peptide may bind to an MHC or be immunogenic or being recognized by an antibody response. Here, we are dealing with tools that operate as a substrate on already defined epitopes to gain additional information or to operate in some fashion on that. And this is the screenshot of the IEDB analysis resource analysis tool tab. And you can see that there are a variety of different tools listed there. You also note that some of the tools have this little lab flask next to it. What it means that these tools that have the lab annotation are tools that are not fully and completely validated and optimized. We strive to, and we do that on purpose, to put them out even though they're not completely finalized for two reasons. One, if we don't want to hold back on making the tool available. And second, we hope to gain some additional insights by users on how the tool works and perhaps how it could be improved. So forth. So let's take a look at a few representative tools from this menu of analysis tools and start with population coverage. This is a fairly common occurrence where someone has a set of epitopes and knows which particular HLA alleles this epitopes bind to or perhaps are recognized in the context of, and wants to know in the general population what fraction of the population is capable of recognizing at least one epitope, or how many epitopes on average the population recognizes. Furthermore, as you probably know, HLA molecules vary in frequency in different ethnic groups and different ethnicities. And so one could ask the question, is this set of epitopes, what is the population coverage in ethnicity A versus ethnicity B? So we use the HLA genotypic frequencies that are found in allelefrequencies.net, and then do basically calculations based on what is the classic genetic Hardy-Weinberg equations to know how frequently this particular epitopes are recognized. So, for example, let's imagine that we have a set of 11 MHC class 2, HLA class 2 restricted epitopes. And so for each of these different epitopes down here, we know what alleles do they bind or are restricted to. And we want to know what is the population coverage. We can select different parameters, but the main thing is here we are interested in coverage in the North Africa populations. So we select that, we enter our set of epitopes and their associated restriction, and then we hit return, and that the data is that is returned is shown in different formats. And one is in this table format where you see for the different population or areas that we selected is shown the coverage. And this means what fraction of the general population in that area would be expected to recognize at least one of these 11 epitopes. And you see that on average, it's about 76% of a different population in these different areas would recognize at least one epitope. Another way to look at it is, okay, we have 11 epitopes. So on average, how many epitopes the average individual would recognize? And you see that they recognize about 7 different epitopes. That is because these 11 peptide epitopes that we used are promiscuous and are capable to bind in more than one HLA molecule. So actually it would be quite a laborious calculation to do it by hand, believe me. And this other graphic form shows essentially the same format, shows how many epitopes are recognized by at least 1 or 2, 3, 4, 5, 6, and so forth epitopes, and this is the average and histogram and so forth. Another tool here is this epitope conservation tool that calculates, can seem to be able to get back. It calculates the degree of conservancy. So this is another situation where, say, you have a number of different epitopes, and you want to know how conserved they are. So these days we can talk about variants, for example. So you have a bunch of different SARS-CoV-2 epitopes, and you have a sequence of a bunch of variants, and you want to know how well conserved this set of epitopes is, how well conserved is in the various different sequences of interest. And obviously, conservation is dependent. The concept of conservation depends on setting a threshold. What do you consider conserved? So you may ask, I want to know how many epitopes are 100% conserved or 70% conserved, because it's known that a different level of sequence homology may actually still be conducive to cross-reactivity at the level of T cells, for example. So in this particular case, what we would do is enter the sequence of a test epitopes, the epitopes of interest. In this particular case, this is a Lassa nucleoprotein derived epitope. And then we would enter a bunch of different Lassa fever virus nucleoprotein sequences, which are the ones we want to know how well conserved the different epitopes are in this particular sequences. And so then when you run this the tool, you would return this kind of data. So here are the different epitopes that we are investigating. So nucleoprotein 421, for example, that has this sequence, is 15 amino acids long. And then we have entered 64 different NP Lassa fever protein. And the tool will tell you that this particular epitope happens to be 100% conserved in 63 out of 64 sequences that we enter, corresponding to a 98% rate of being 100% conserved in the taste set. We can also click on a particular epitope, and that will give me the breakdown for this particular epitope of all the different sequences in the different subsequences in the different Lassa nucleoprotein sequences, and will also highlight what are the changes that are seen in case the particular epitope is less than 100% conserved. This is an epitope clustering tool, and this is also an occurrence that is found in frequently in analyzing epitope data. And some of this was alluded earlier on in the sense of the clustering and the immunobrowser tool, in the sense that what happens often is that people study the same region, but they don't make necessarily the same exact peptide. They make maybe a peptide that is a couple of amino acids longer or shorter or shifted and so forth. So how do we deal with that? How can we kind of cluster sequences that are similar? And there are different ways to do it. And I have implemented 3 different approaches because in different situations, each one may be of use. The first approach essentially connects all peptides that have a certain degree of homology in a cluster. And so, for example, we can say, I want to connect all epitopes that have 70% homology. And the problem with that is potentially that you can have, say, that the first peptide has 70% homology to the second one has 30% homology to the third one, and the third one has 70% homology to the fourth one, and then the first and the fourth one have nothing in common. Indeed, for example, if you think about overlapping peptides, 15 mers overlapping by 10, they all have 70% homology. So you would have a whole set of overlapping peptides spanning an entire protein classified as a single cluster. So that obviously is not an optimal method for some applications. The other way to do that is to say, no, I just want to have a cluster in which each one of the peptides have 70% homology to every other peptide included in the cluster. That is more stringent, but also it can lead to a situation where the same peptide is found in multiple clusters. To kind of overcome these 2 potential drawbacks, we developed this cluster break method, which is an extension of the first approach, but also has original ways to deal with the extended problem of having clusters that are really not clusters. So here's this example. I just wrote down some sequences here: trapper, clapper, snapper, wrapper time, happy days, hippie, payday, calamari. This is a sequence of a very well-known over 3 to 3 IEDB restrictor peptide, just for fun. And so we enter all the sequences that we want to cluster. We went specify parameters, what kind of clustering threshold, homology threshold we want to use to cluster. Then we click go to step 3 and we select our algorithm. And so if we select all the connected peptides in a cluster, what you will have is this type of results. And so you see the issue that I was mentioning earlier. So it clusters things that have some degree of homology, but in the end, Trapper and Timeless really don't have 80% homology at all to each other, but they are linked by a series of peptides, each one of them having 80% homology to the next one. If we instead click on the fully interconnected cluster method, you will have a finer grain of clusters, because now everybody has to have 80% homology to all the members of the cluster, but you will find that the same peptide is also can be found in more than one cluster. The cluster break method instead will generate, again, relatively larger clusters, but they are connected, and you can see how connected they are, and basically it separates these clusters in subclusters that have more tight homology to each other. Another tool that I would like to present to you briefly is the rate tool. And this is a tool to infer restriction. And it's not meant to provide an absolute crystal clear restriction, but rather to analyze the data, some more information, suggest what are the likely HLA restrictions. And so the methodology here is really based on the same methodology that is used to determine, for example, if a certain gene or a certain HLA allele is associated with a particular disease. And basically, what you do in those situations is you say, oh, this particular DQ3.1 HLA molecule is associated with diabetes. And the association means that the disease is much more frequent in people that carry that gene as opposed to people that don't. And it's calculated a certain risk, odd ratio risk factor. And so here we're applying the same concept to determining HLA restriction based on the concept that if a particular peptide is restricted by, say, HLA-DR4, and we test this peptide in a population of 100 people, then we will see that the responses are linked to people that have DR4 as opposed to people that do not express that allele. So what we do, we need to provide the data. So we need to provide the population in which this peptides were tested and the response data. So, for example, here we have a fictional population of different donors, and you see this is the HLA molecules, the list of the HLA molecules that each donor expresses. And then we have a list of, again, fictionary peptides that have been tested in this donor population. And we know that, for example, this particular peptide was recognized by donor 7, or this particular peptide was recognized by donor 2, and so forth. And obviously, this works well, or works best the more data you have. And if you have a relatively large data set with a good population size, you can actually get some interesting results. And so this is the formula that are used to define odds ratio or relative frequency. It's explained in more detail in the help section of this tool, but essentially, again, it contrasts the frequency of different HLA alleles in responders versus non-responders to one particular peptide. And so we hit submit, and we can choose different formatting for reports, a more synthetic report or a more complete report. And here is the report with all epitopes, listing all the different epitopes, and this is and different alleles. And this is a more condensed report where we have, based on our experience, set thresholds of things that we think are more likely to be of interest. So, of course, in the case of HLA restriction, you're looking for a positive correlation. So you don't necessarily want to look into negative correlation, and also selecting a certain p-value. And this, I want to emphasize that it's not Bonferroni corrected. So again, this is not meant to be a definitive answer, but it's often a way to narrow down, to flag some likely restriction. And so, for example, in this particular case, we have this particular peptide. And when you look at this particular HLA allele, you will see that people that had this HLA allele, there were 6 people in this population that had this allele and responded, while in no case there was someone that did not have this allele but responded. Conversely, there were 18 cases of people that had this allele but did not respond, and this is a common situation in the sense that someone that has a particular HLA allele doesn't necessarily mean that they would recognize that peptide. And then you had also 26 people that did not respond and did not have that allele. And so you can calculate, basically on the basis of a Fisher p-value, what is the likelihood that this would occur by chance, and you have this p-value that is listed next to the peptide infer potential restriction combination. Another tool is the T-cell epitope immunogenicity prediction tool. And there are different tools here. One is the CD4 immunogenicity prediction, one is the deimmunization. I'm just gonna demonstrate a little bit what the deimmunization tool does. So what the deimmunization tool was designed to answer is a relatively common situation where, in the context of development of protein drugs, one has a protein which is either a wild type or engineered that is injected into patients as a drug. Monoclonal antibodies, hormones, or other optimized proteins are a common example. And oftentimes, some individuals develop an antibody and immunogenicity reaction against the drug. If it's an antibody response, must be mediated by a CD4 T-cell response. And so there is interest to identify these potential problems and perhaps removing or decreasing the likelihood of this unwanted immunogenicity by identifying peptides that bind well and introducing substitutions to decrease the binding affinity. So what the tool does is scan a protein of interest provided from the user and finds what regions have a high likelihood of being immunogenic because are capable of binding to multiple different HLA molecules, and then suggests substitutions that could be of interest because are predicted to decrease the overall binding propensity of that region. And also consider effect on substituting of substitution on neighboring peptides, because what can happen is that you introduce a substitution that decreases the binding affinity to the alleged potential epitope, but simultaneously creates a new epitope to the N-terminus or to the C-terminus of that sequence. So in this particular example, for example, we enter EPO. EPO is a drug that has been linked to severe side effects due to induced immunogenicity. We select a threshold. We run the algorithm. We find that there are 4 different potential regions that maybe have a high predicted propensity of binding HLA class 2 molecules. And then you can run the algorithm right there. We suggest you put your email address because sometimes the calculations are very intensive and takes a while to get the results. And so we select the peptide we want to analyze. There is a code here that goes from good to bad in terms of 1 being good and 10 being the worst, in terms of the predicted effect on the neighboring regions of a substitution introduced. There are 2 neighboring peptides, and so we need to worry about 2 neighboring peptides. The best scenario is, of course, if the peptide is drug itself, it's a 15-mer, or it's at the N-terminus or C-terminus of a protein, so it will not have neighboring peptides, or at least have only 1 set of neighboring peptides. Or other situations in which is the substitution introduced is predicted to have no effect or even a decrease and so forth. So this is how the outcome of the output of the algorithm looks. So the wild type is there, and then there's a bunch of different substitutions that are proposed and suggested by the algorithm with the predicted effect on the neighboring peptides, both at the C-terminus and N-terminus. And here must be emphasized that these are substitutions that are predicted to decrease binding affinity. But at this point, one needs to also work with a medicinal chemist, because the substitution may be decreasing binding capacity, but may also be affecting the biological activity of the protein. So obviously it doesn't do you any good to have a drug that is non-immunogenic, but is also not bioactive. So just to summarize, these are the different tools that we went through: the population coverage to estimate what coverage one or more epitopes may provide in a particular ethnic group or in a general population, the degree of conservancy of different epitopes, how to cluster different epitope sequences or peptide sequences by different methodology, how to infer potential HLA restriction based on known immunogenicity data from a peptide set, and how to propose different single substitutions to decrease potential immunogenicity. And with that, I think I will stop and we can go, I believe, to the Q&A.
Yes, thank you so much, Alex. Thank you for the presentation on analysis tools. And we are running about 10 minutes ahead of schedule, but again, that just means more questions can be answered, which is great. So at this point, I would like to invite Bjorn and Zeynep back to the virtual stage to kick us off with some questions. And I think Zeynep, you presented first, if you'd like to take the first question.
Yeah, so the question, so there's no cancer-specific question, but there's one question regarding the prediction tools, which is the results of the DMHC-PAN BA method shows the IC50 value and the EL shows percentile rank values. What is the best way to choose the epitopes based on the IC50 or percentile rank? So the BA methods have been trained on binding affinity, and the EL method has been trained on eluted ligand data. So our internal benchmarks across different data sets have shown that the EL method is always more accurate than the BA method in predicting epitopes. So that is also why the EL method is the recommended method right now in the T-cell tool. Yeah, so we are also providing thresholds to use. So as a percentile rank, 2.0 would be a good threshold to narrow down the epitope candidates. Yeah, I hope that answered the question.
Thanks, Zeynep. I'll take, I started and grabbed a few of the questions. So let me start with the first one from Taina, which was, are there official Docker images of IEDB tools? Yes, there is like a big Docker image that is distributed. If you're going through the download link, there's one to get to the whole image of all the IEDB tools. And that is actually in the Docker file that allows you to get essentially like a virtual image of the entire server. There's not tool by tool Docker images. We are actually doing that. We're experimenting with that, but it's not something that we are currently making available publicly, but I think we are moving there. It seems to be, I mean, obviously, it's been the way the field is moving. And let me actually put a second question because I grabbed too many of them. Dear Dr. Peters, what's the prediction accuracy of the IEDB class 1 immunogenicity? So tomorrow we're going to go into more details for all of these questions, specifically on accuracy. But I can tell you right now for the immunogenicity prediction, which goes beyond the binding prediction itself, it provides an additional maybe 10% or so increase in accuracy if you add it to binding affinity predictions. But the binding affinity predictions are really what allows you to narrow down from a set of whatever, of like 1,000 epitopes candidates to the 100 that are going to be containing all the epitopes. And with that, I'll stop. Alex, you want to take the next one?
Sure. So the question is, does the number of ethnic groups affect the percent of population coverage? For example, when I did population coverage for my epitope set and for the IEDB example for the UK, one ethnicity, it result population coverage different from Brazil, many ethnicities. Not necessarily. So the population coverage is really not predictable a priori. And let me explain a little bit more. So first of all, it's first and last is dependent on the specific binding capacity of the particular peptide set you use. So to make a case in point, say that you have defined a set of epitopes in Brazil. I would expect that maybe that population coverage will be great in Brazil because specifically binds to whatever alleles were found there. And if you go to the UK, you might take a hit because the HLA molecules might be different. Conversely, if you have a set of epitope that was defined in the UK, you might get a lower coverage when you go to Brazil. Another point to, and so in general, to have a more diverse set of ethnicity doesn't necessarily mean that you will have higher coverage. Now, another point to emphasize in here is that there is a tremendous bias in the data that is available in the literature towards alleles that are frequency in Caucasians. And that's because people have historically done a lot of experiments in Caucasian. For example, in the case of HLA class 1, everybody studies HLA A201, which happens to be very frequently in Caucasians, but less so in other ethnicities. Now, the tool does obviously do whatever it can do because the tool doesn't know if that particular peptide is also restricted by other alleles. But for example, if you recall, when we went through the examples in the database, I pulled out the HBV core 18 to 27, which exactly was an epitope defined in A201 individuals. But when people started studying epitope in different ethnicity, for example, we started studying it in Asia, where A201 is not as common, but other alleles, A2.2, A2.3, A2.5, A2.6 are more common. It turned out that the same peptide was also recognized in the context of these other variants that are less Caucasians and more Asian of HLA-A2. And so that the population coverage provided by the tool is often just an underestimate, it's a minimum estimate based on what is known. But if people would do more work in different ethnic groups, it would be a more reliable estimate. Who goes next?
I think I got the next one, Alex, from I. Simpson. If an epitope is identified as immunogenic in the IEDB database, will it always be predicted to be immunogenic in the IEDB analysis resource tools? So the answer to that is no. The tools are not directly linked up with the database itself. That's why I recommended, like, if you have like a set of questions that you want to ask for tools, to first start by asking, is there information available on the database? So it would actually be nice, and it has been asked before, to make that an automatic link, so that if you're sending something through the tools and a sequence is in there that has already been experimentally identified and characterized, to give those results there automatically. But currently that's not the case. So we are relying on our users to try the database first and then go to the tools. I hope that answered the question. I think that's the last question we had in the Q&A.
Yes, I think so too. So I think we will thank you, Zeynep, Alex, and Bjorn, for your presentations and for your time for the Q&A. I will slowly move into the conclusion, and then if there are any other last questions that pop up, we'll answer them in the optional Q&A section. So thank you, Bjorn, Alex, and Zeynep.